unfortunately today we discuss about the polymerase, DNA dependent DNA polymerase, prokaryotic and eukaryotic DNA polymerase, DNA dependent RNA polymerase, RNA polymerase and DNA primers. There are different types like DNA polymerases, RNA dependent DNA polymerases, reverse transcriptase, RNA dependent RNA polymerase, that is RNA replicase. So this RNA replicase showed RNA synthesis even the presence of the actinomycin that inhibits the DNA primed by the RNA synthesis. Like Baltimore and Franklin showed the viral productions with the specific cytoplasmic fractions. Those RNA primed RNA synthesis was demonstrated. This enzyme has been named as RNA dependent RNA polymerase or RNA replicase. All RNA viruses except retrovirus encode a protein since the host dependent not possess it. Structure of QB replicase, 4 subunits like 3 provided by E. coli host on one viral RNA genome. The synthesis of in 5' to 3' directions, lack of proofreading functions and requires the RNA template and specific RNA in the viruses. RNA replicase was isolated by the Spin et al. From the QB bacteria. This requires the RNA template magnesium RNA NTPs. QB replicase is a tetramer consisting of 1 replic molecules of 3 host proteins. Replicase copies viral plus standard generated negative strand. During the synthesis of negative strand is not contacted with the positive strand only. At the polymerization site, initiations of several strands occurs before the first is complete and the replicative from the branch. The negative strands are released and immediately used by the replicase to form positive strands only. Reverse transcriptase, so extensions of the central dogma of molecular biology like DNA to DNA synthesis is replication, DNA to RNA is synthesis of transcription, RNA to protein is translation, RNA to DNA is reverse transcription, RNA to RNA is RNA replications. The Tiemann-Betlman independently demonstrated the presence of reverse transcription in several ovine and mouse returns. So RT and RNA dependent DNA polymerase, it is used in recombinant DNA technology to transcribe the mRNA into the double-stranded DNA. So reverse transcriptase has 3 enzymatic actions. It copies an RNA molecules to yields DNA double-stranded DNA RNA using a primer and joining DNTPs 3' to 5' linkages. It copies a primed single-stranded DNA to form the double-stranded DNA. Ultimately, it degrades DNA to RNA or hybrid. This is called as RNase activities. So structure how the gene expression, so gene product, how this transcription and translation, that's GAG and poly envelope. This is the host DNA. Then here is segregation, bifurcation like translocations like the polypeptide. And here is the viral structural protein. Ultimately, finally releases of viral enveloped proteins here released. Then core of bacteria phases like this glycoprotein and MHC glycoprotein 120 like the core protein. Some of these the fine layers. So retrovirus, how this viral genome is segregated, so then RNA degraded, then DNA circulations like a viral consumption, then all viral translated viral enzymes. So ultimately viral membrane proteins transported host cells. So final viral assemble and budding that takes place. So how this procedure will be formed of these things about the thank you DNA polymerase. Thank you and thank you very much.